Welcome back to The Explainer, everyone. I'm so pumped you're here today because we're diving straight into the invisible, high-stakes world of veterinary microbiology. We're looking at some of the absolute toughest, most indestructible microscopic threats known to science, and more importantly, exactly how diagnostic experts manage to track them down. Imagine trying to hunt down a microscopic fugitive. But the catch? This fugitive has locked itself inside an impenetrable biological bunker. Standard diagnostic tools, normal laboratory dyes, they just bounce right off. You literally can't see it, you can't easily kill it, but you absolutely need to identify it to save a patient. So how on earth do you spot a deadly microbe wearing biological armor? Well, that's exactly the puzzle we're cracking today. Okay, let's jump right into this. Here's our game plan. First, we'll look at the biological bunker itself. Then, cracking the spore armor, followed by the Schaeffer-Fulton method. After that, interpreting microscopic clues. And finally, we'll wrap up with the real-world veterinary stakes.
Starting off with part 1, the biological bunker.
The technical term for this bunker is the endospore. Now, what's really fascinating here is that word dormant. You see, in this state, an endospore isn't a living, breathing, multiplying cell. Not at all. It's a highly resistant, dormant structure that certain bacteria form when their environment turns downright hostile. Think of it like the ultimate microscopic panic room. When things get bad, the bacterium just packs up its essential DNA, builds a literal fortress around it, and goes to sleep. So what actually counts as a bad environment for a bug like this? These unfavorable conditions triggering spores can be things like nutrient depletion, which is literally starving to death. But it goes way beyond just running out of food. These spores pop up in response to intense heat stress, extreme dryness, and even nasty chemical exposure. The bacterium basically shuts everything down, builds its bunker, and just waits. It waits it out until conditions are favorable enough to germinate again. It's an incredible, honestly kind of terrifying survival mechanism. Which brings us to part 2, cracking the spore armor. Now, if this bunker is built to shrug off extreme heat and deadly chemicals, how do we even get inside? Well, here's our main obstacle. Endospores are wrapped up in a thick, keratin-like spore coat. And keratin? That's the exact same tough stuff making up animal horns and hooves. Because of this heavy-duty armor, your ordinary staining techniques are totally useless. You drop a normal stain onto a slide with these guys, and it just washes right off. So to crack this armor, we're going to need a very specific physical and chemical intervention. But before we breach the hull, let's talk about our goal. If we pull this off, we want to see a really vivid contrast. We want these indestructible endospores to absorb a primary stain and turn brilliant green. At the same time, we want the normal, active, vulnerable bacteria, what we call vegetative cells, to turn red or pink. Green means danger, red means standard cells. That's the visual endgame we are chasing here. And this brilliantly illustrates part 3. The Schaeffer-Fulton method. Let's walk through this genius lab procedure. Once you've got your bacterial smear prepared, step 1 is flooding it with our primary stain, Malachite green. Then, step 2, and this is the secret weapon, heat. You gently heat the slide until steam rises for 5 solid minutes. This literally forces the green dye right through that tough keratin armor. Once the dye is in, step 3 is a quick wash with distilled water. The water strips the green dye out of the normal vegetative cells, but those armored spores, they lock the green inside. Then, step 4, to actually see our now invisible normal cells, we use a red counterstain called Safranin for 1 minute. Finally, step 5, wash and blot dry, and step 6, you observe the microscopic battlefield under 100x oil immersion. But listen, as a microscopic detective, you've got to be careful, because things can absolutely go wrong. What are our main sources of error here? Insufficient heating is the big one. If you don't steam it enough, that green dye is never breaching the armor. A thick smear, that'll just clump all your cells together, making observation completely impossible. Using old or poor stain gives you super faded results. And overwashing can just ruin the delicate balance of your dyes. You've got to be precise. Thin smears, proper continuous steaming, and exact timing are the only ways to guarantee this works. Moving right along to part 4, interpreting microscopic clues. All right, so we've followed the protocol flawlessly. What are we actually looking at? The crucial point is that we aren't just looking for green spores on the slide. We're looking at exactly where they are located inside the red vegetative cell. These positions act just like microscopic fingerprints.
A spore might be central, sitting smack in the middle. It might be subterminal, kind of near one of the ends. Or it could be terminal, parked all the way at the very tip of the cell.
Let's see how these positional clues play out with two pretty notorious killers. Say you look into the microscope and you see large red rods with green spores sitting perfectly in the center, those central spores. You're very likely looking at Bacillus anthracis. On the flip side, if you see a green spore bulging right at the very end of a cell, a terminal spore, it gives the whole thing this highly distinct drumstick look. And that drumstick? That's the classic calling card of Clostridium tetani. Just by noting the position of that tiny green dot, we can instantly begin identifying the pathogen. Finally, Part 5, the real-world veterinary stakes. So, why does a 10-minute staining protocol matter so much outside the lab? It matters because those little green dots represent some of the most devastating livestock and poultry diseases on the planet. I mean, Bacillus anthracus, that causes anthrax. Clostridium tetani causes tetanus. We're also talking about enterotoxemia from Clostridium perfringens and botulism from Clostridium botulinum. These diseases can wipe out agricultural populations incredibly fast. Our source material really hits the nail on the head here, stating, endospore staining is important for diagnosis of diseases caused by spore-forming bacteria, important in veterinary diagnostic labs. You see, the Schaeffer-Fulton method isn't just some abstract academic exercise. Because it quickly differentiates these armored spore formers from other normal bacteria, it's an absolutely essential tool for preliminary diagnosis. It's quite literally the first line of defense in veterinary medicine, letting experts see an unseen threat and take action fast. We've seen just how incredible this microscopic armor really is, and how brilliantly scientists have figured out how to bypass it using basically just heat and some green dye. But I want to leave you with this final kind of chilling thought. If endospores are a bacterium's ultimate survival tool, capable of withstanding extreme heat, dryness, and chemical attacks for years, what else is waiting dormant in the dirt right now, just waiting for the right conditions to wake up? Food for thought, right? Thank you so much for joining me on this explainer. Keep asking questions, and I'll catch you next time.